So as Steph mentioned, so the project is called ArHa. And what I wanted to do is I wanted to produce a data set where we would be able to synthesize all of the literature, all of the published assays that have been conducted in small mammals for two viral families, the Arena viruses and the Hantaviruses. And I put in a project where I was going to start looking at sort of the spatial sampling biases and things like that, but we haven't got to the point where I've been able to do that. We're still at the database construction stage. So this will be sort of talking a little bit about where we've got to so far and sort of our plans for the next step. Even as far as I have, because of the help from a couple of Steph's lab members, Ricardo and Grant, who's an undergrad who works with her. And more recently, Dave Redding and his team from the Natural History Museum have also got involved and started helping out with some data extraction. So I just want to introduce a little bit about what the arenaviruses and hantaviruses are. So they're both viral pathogens that occur in multiple species. But the sort of the zoonotic ones, the ones that are of particular interest to us from a public health perspective, are those that tend to come from rodents or other small mammals such as shrews. They're globally distributed, so either the hosts exist pretty much everywhere or the pathogens themselves have been detected in a lot of countries as well. But within them, there's a subset that are of zoonotic concern. So there's a lot of arenaviruses and hantaviruses that we don't expect to cause any problems if a human were to be infected with them, or whether they can even infect people. But for example, with the arenaviruses, the ones that we're particularly interested in are Lassa fever, which is the pathogen that I studied as part of my PhD and is the main work of my postdoc. And so this is a viral hemorrhagic fever that's limited to West Africa. But then there's the New World arenaviruses, such as Hunin virus in Argentina as well. And then on the hantavirus side, we have sort of the older world hantaviruses, namely Pumla and Hantan virus, and these cause hemorrhagic fever with renal syndrome, which, despite the very scary name, is a relatively mild disease. And so most people that get infected will not have a severe illness. They may need some sort of supportive care, but the mortality associated with that is very low. However, in the US, where Sin nombre virus is a hantavirus, there the mortality associated with detecting a case is very high. It may be that there's lots of people that get infected, but that don't come into contact with healthcare. And so a lot of this, similarly to what people were talking about previously, exists in like a data sparse space. But we would consider that these pathogens should be of some degree of interest to people all over the world, hence these very filled global maps. So I really got interested in sort of this idea and also working with Arena because of some work that Rory and the others have done with the Clover data set, where they synthesized host pathogen interactions across viruses, parasites, and bacteria to produce their data set, which is amazing. However, it's a little bit limited in that there isn't any sort of geographic or temporal data associated with the associations that we see between hosts and their pathogens, which can limit some of the inference that you want to then go and do with these sorts of fantastic data sets. And to get sort of more contextual information about how those tests were conducted or the populations that they were studied within, you needed to go to the underlying publications and sort of read up on them. And so I thought that having something where we try and bring it all together, and that it's a linked data set that goes from sampling the host in their natural environment to sort of identification of the pathogens, and then, if available, the sequencing data as well. And that would give us a very rich data set that we can ask lots of different questions on. But something else that I wanted to, sorry, my internet connection is unstable. I hope I'm not talking to the air. Another thing that I'm interested in is sampling effort and how these biases can be propagated through our analyses and accounted for in some of the models that we use. So, for example, in phylogenetics, there's ways, or there's developing ways in which you can incorporate unsequenced samples to integrate into your phylogenetic analysis. So the specific aims of this were to produce a geographically and temporally rich data set of small mammal sampling for these two pathogen families. Ultimately, I would like to expand this to be all viruses and then all bacterial zoonoses of rodents or other small mammals and all parasites, but that's getting ahead of myself. And to link to the data, the sequence data that we have on GenBank, but to be able to then enrich the metadata. So I don't know how many of you have experience of trying to use some of the metadata on GenBank, but it's quite patchy. So sometimes they will have the host name that the pathogen was sequenced from, but then they may not have the location, they may not have when it was sequenced, and they definitely don't have how many animals they sampled in order to find that pathogen. And so this would allow us to do that. And as I mentioned earlier, looking at sort of geographic and temporal sampling biases and how we can integrate those into downstream analyses. And just so that it's not just me talking about producing a data set, I can give you some nuggets of some of the ideas that we would like to use this for. So the question that Ricardo, I think, is going to be focusing quite a bit on for his PhD is looking at host overlap. So either looking at sort of the ecological niches of the hosts or just purely sort of locations in which they've been detected or not detected, looking at those in a sort of spatial overlap aspect and looking at opportunities for viral reassortment, using his sort of bioinformatic and phylogenetic skills, tying into the ecological data that we're producing from this data set. And then looking at, so arenaviruses and hantaviruses generally have been thought about as like one host diseases, one host pathogens. That's becoming a little bit more questioned recently. So can we look at some of these drivers of cross-species transmission and looking at maybe sort of any factors of adaptation among the viral genomes for those that are more or less likely to cross-species barriers? One of the questions that sort of brought Dave Redding and his team in more is looking at the sort of the estimates of pathogen prevalence within a species. So is there evidence that hosts of a species are more likely to be infected if they come from the more ecologically viable settings? This abundant center hypothesis sort of translated from individual animals to sort of their pathogens as well. And using this sort of data as well to ask questions about host traits. So bringing in some of the sampling bias questions to sort of enrich our understanding of whether certain host traits are factors in cross-disease transmission across species or other factors. So this is how we've gone about this. Started off with a very basic, simple search term, anything that mentioned rodent or shrew, and anything that mentioned arenavirus or hantavirus, but these were all expanded terms. So, for example, rodent includes all the species of rodents, shrew includes all the species of shrew. We have a very loose inclusion criteria, which has meant that unlike Lambo's talk earlier with maybe 6 studies relevant, we've got 917 just on the initial screening stage at the moment. And that was narrowed down from nearly 3,000 after deduplication. And oh, I missed that one out. Currently, we've extracted data on around 236 studies. But as I mentioned, a few different people are helping with this, and I've only gone through the cleaning steps on the data that I've extracted. So I'm just going to talk about all of the data that I'm going to present now is just related to those 110 sample studies that I've extracted data from myself. And that represents 12 of our total sample that we need to get through. But that's around half of all the ones that we have data on. Similarly to every other time we see maps, certain countries are more likely to publish studies on sampling pathogens in animals. So the US, it's leading the way in bright yellow. China has a fairly large burden of hantavirus disease. Also, it's got a very large population. So, and they've got fantastic data sets, but there's quite a lot of publications that have come from China and similarly to Russia. So at the point that I was making this presentation, we have around 36,000 individual records for small mammals. And those aren't at the individual level, but they're at a geographic location and temporal sampling point resolution. Different studies have different resolution. That's something that we've been dealing with throughout the project. But it represents around 140,000 small mammal detections. And again, some of those are going to be capture mark recapture. So again, don't quite relate to specific individuals, but the majority of them are individual rodents that have been killed after sampling. And that comes from around 1,400 distinct sampling locations, which go from any resolution of like a specific trap or a specific grid square, all the way through to sampling across the country could be counted as one location, depending on how it's been reported in the publication. We have similarly 40-something thousand pathogen records, and that's because you may be testing a single individual for multiple pathogens, particularly with some of the hantaviruses where the assays aren't that specific. You may have a general sort of screening ELISA for hantaviruses, but then you may do more specific tests for a particular viral pathogen that you're interested in. And out of those 44,000, we have over 200,000 individual pathogen assays, and around 6% of them are positive. So the vast majority of samples are negative, which is fine. We expect that. But it's quite a high positivity. And so we probably need to look into some ways about looking at sort of reporting bias as well in this. In terms of linking to GenBank data, so we can link to 1,600 sequences, and these are based on accession numbers or accession, whatever we call them if they're not numbers, accessions in a publication. And just over 1,000 of those are the pathogen sequences that we can link back to the host in which they came from. And we also are able to identify some host sequences. Most of these are just things like mitochondrial DNA rather than like the sequence of a specific individual, but there may be some things that we can do looking at sort of like contact reconstruction through mitochondrial DNA, but that's one of my pipe dreams rather than anything feasible at the moment. So I thought I'd just show some more maps. So this is a map of the assays for different hantaviruses. So the more yellow, more green, it just means the more they are. So there's lots of sampling in Montana, California, and Nevada in the US, and that's related to sort of looking for sin nombre, and a lot come from sort of Central and Northern Europe, and that's looking for like Pumalovirus, which is another hantavirus. But we have very little sampling so far from Sub-Saharan Africa, pretty much none from India. And this is despite from that first one of those first plots that I showed you, that we know that there's hosts for potential zoonotic hantaviruses within those locations, but the research effort that's been put into looking for it is fairly minimal for most of them. Arena viruses are less tested for. No one wants to look for them in the US. Possibly the only one that's of interest there is a very long name, lymphochorion. It causes a type of meningitis if a newborn gets it, but other than that, it doesn't cause disease in people. So there's not much motivation to testing for arenaviruses there. But in Sub-Saharan Africa, where we know there's Lassa, we're more interested in looking for that as well. One of the headaches has been sort of trying to harmonize these data. So anyone that's gone and just done any sort of systematic review knows that people write about things in different ways, even down to the species of rodent that was sampled or the pathogen that they were looking for. So one of the things that we've been trying to do is thinking about how much processing of the data we want to do while producing the data set and how much of it we actually want to do once the data has been extracted. So I've just been asking people just to extract the data as it's reported in the paper, and then we'll deal with all of this stuff. And I just wanted to highlight this tax size package just in case anyone else comes across these problems. It's a very nice way of being able to do like fuzzy matching on free text and then assigning it to a unique identifier. I use GBIF, but there's other taxonomy platforms that you can use. And then that allows other people to then translate that into however they want to use their taxonomies. And similar thing with pathogen taxa, but I've not found a sort of a good resource for that. So I've just been using the NCBI taxonomy browser, but maybe someone has a suggestion for like a useful pathogen taxonomy to use. And that's just me searching for the name and then just pulling out what the true viral name is, I suppose. What I'm also doing is I'm trying to enrich the data in the sense of we can impute non-detections of hosts based on trapping data. So if a study has reported finding, say, Rattus rattus in one location, but then they've not reported its absence in another area, the fact that they reported it elsewhere means that I feel somewhat comfortable saying that that means they didn't find it somewhere else. So again, trying to get as much out of the data as we can currently. And then I've got a few things on my to-do list. So one of the things that I'm interested in is thinking about this coordinate resolution. So some studies report exact locations of where the traps are. Others say, okay, we trapped in this state, but trying to feed that forward so that other people can use that later on in an analysis, I think is quite challenging because states are different size depending on which country you're in. And then there's lots of these different administrative levels. So just trying to allow people to have as much data and then pick and choose the things that they're interested in. I thought I'd show some host pathogen associations because that's sort of what got me into this to begin with. This is a very small graph, so I don't know if you can zoom in, but just the takeaway is this idea that you have one host for a pathogen may not be that true. So for example, for Sin nombre, there's 20 different species that have been found to be infected. It doesn't necessarily mean they're a host, obviously, but it does mean that maybe we want to think about the role that they may be playing in sort of maintenance of the pathogen among the host population. Similarly with like ArHa, like 12, 14 different species have been detected. And the lighter colors are more samples assayed. And so this is very skewed. So there's some pathogens that have a huge amount of effort and others less. Another very messy graph, just trying to understand this idea that how do we assess the prevalence of a pathogen in a population. So for example, for Sin nombre in Peromyscus, these purple dots, studies have reported anywhere, or sampling events have reported anywhere between 0% and 90% pathogen prevalence. And so how do we use some of this data to inform our understanding of disease transmission or pathogen transmission? I won't do this because we're running out of time. And yeah, so there's a lot of code that people are very welcome to sort of repurpose. We have, I'm trying to sort of get out the protocol paper for this, just explaining our thinking and how we extracted the data. The Pharos data harmonization approach sort of informed a lot of this. So I think that's quite a nice sort of follow on from that. And the clean data is also available on the GitHub. It's not complete, but it is for the 110 studies so far. So if anyone wants to have a look at the data, it's all available. And there's an app which I just updated today, and it should work, which can be used to sort of explore it in a bit more depth if any of this has got you interested. And I will end there.